Pleasure today to introduce our speaker, and his name is Joseph Sweeney, and he's a research fellow in the School of Biosystems Engineering and Food Science. His expertise is in the area of E. coli-based biosensor development, and his group are currently developing biosensor-based monitoring and control systems, particularly around the area of green biorefineries. Working on projects Life Farm for More and AgriFine. So welcome, Joseph, or Joe, and we look forward to hearing all about your work. Just to note to everybody, if you have questions, we'll come to them at the end, but do type them into the chat at any time point, and we'll come back to those at the end, or we'll turn on microphones at the end and have a more informal chat. And answer any questions that we have at that time point. So just feel free to interact whatever way you're comfortable with. So I'll hand over to you now, Joe. Thank you very much.
Thanks everyone for joining me today. I'll just begin with maybe just giving a background of how I came up with the idea of these biosensors, these whole cell biosensors. So in 2012, I began a PhD. It was on the Charles Parsons Energy Research Program in the School of Biosystems and Food Engineering. I was very fortunate that the Charles Parsons research program was kind of in its latter period, and there was a number of PhD topics that came available, and just something needed to be done in the area of anaerobic digestion. So I was given a very broad PhD topic. So kind of the technical definition would have been exploring methodologies, technologies, and strategies that can be applied to improve anaerobic digestion. And after doing an extensive literature review, kind of identified that real-time automated monitoring control systems are lacking in anaerobic digestion. So I had a primary degree in microbiology and a master's in computer science at this time. So by combining those two together, I came up with the idea of developing a real-time monitoring control system for high throughput grass silage bed ADs using E. coli, using whole cell biosensors. So I'll get into a little bit more why particularly E. coli, and then also why this high throughput grass silage bed AD. So as part of the literature review I did, I kind of identified issues with AD. So within anaerobic digestion, you have microbial groupings, which have differing optimal operational parameters. So in essence, it kind of comes down to within an anaerobic digestion process, you have hydrolytic bacteria, and you have the methanogens and acetogens. And actually, I got the pH the wrong way around there. So the methanogens and acetogens, actually like a neutral pH, with the hydrolytic bacteria can operate at lower pHs. So within an anaerobic digestion process, when an overloading event occurs, when this means that there's too much organic inputs substrate pushed into an AD, the hydrolytic phase becomes more dominant. There's a reduction in pH, and what this does is it inhibits the methanogens and acetogens, and this causes a reduction in biogas methane production. And generally speaking, the only recourse you have available to you at this point is to take the AD unit offline to allow the methanogens and acetogens to recover. So because that has an economic consequence if that's done. So the question was at this time, what methods are employed to prevent overloading? And without real-time monitoring control systems, the simplest method that the majority of anaerobic digestion plants apply is they operate them conservatively. They operate them well under the maximum organic loading rate that you can apply. So the question would be then, what methods could be applied to allow for a greater organic loading rate and then maximize the efficiency of the AD process? And a number of papers have proposed if the VFAs, volatile fatty acids, in particular acetate and propionate, could be monitored in real time, then the ratio of acetate to propionate could be an indicator of an overloading event occurring before you see a reduction in methane. And by quantifying the acetate and propionate within an anaerobic digestion process, you could apply real-time monitoring control and optimally operate. So this brings us then Of all the anaerobic digestion processes, why this grass silage fed high throughput AD reactors? So when I started my PhD in 2012, around that time, there was a lot of talk about these new high throughput AD reactors. A lot of the work was done by Jerry Murphy's group in UCC. And what this did was it identified that there's two microbial groupings and it physically separated them within two reactors. So it provided the optimal operational environment to both microbial groupings. You had the hydrolytic bacteria in a dry batch reactor, and you had your methanogens, which are more delicate, and acetogens in this high throughput upward upflow anaerobic sludge blanket. So it's called a UASB. And overall, when the two were combined, you could it was a much more efficient way of maximizing methane outputs from a starting input substrate like grass silage. Now, there is an issue with this, is that still the methanogens and acetogens in this high throughput and sludge blanket, they're quite delicate, and actually the control of the flow into it, this was identified as an issue with these systems, that they needed a mechanism by which they could identify with the control, that they could control the flow of organic loading rate into the second phase. And from that, again, using that acetate to propionate ratio, we began developing whole cell E. coli-based acetate and propionate biosensors. So these whole cell biosensors, what they were built upon was in the 1980s, there was a big development in these biological oxygen demand sensors. A lot of groups in Japan were developing them. And what they did was either take a single microbe or a microbial consortium, and they entrapped it behind a kind of a cellulose acetate membrane on the tip of a dissolved oxygen probe. Very simplistic in its development. And what would happen is when a biological sample is supplied to the cells that are entrapped here, they consume oxygen, and this oxygen consumption could be correlated to biological oxygen demand. So what was the ideal for these microbes or consortia of microorganisms was that you would try and metabolize as many organic acids within your biological sample as possible. So you'd want as many of the individual components within your biological sample to be consumed. And the consumption of those analytes uses oxygen as a terminal electron acceptor. So they would consume oxygen, and you get a better correspondence to the biological oxygen demand in your sample. The advantages were that you could get a reading within 2 minutes. I know a lot of the biological demand kits, the time to take days to provide that reading. They're cheap. You just grow up your cells in a flask, extract them, and entrap them here at the tip of the probe. Cells do not cost a lot of money to generate. They're very robust. They could last 1 to 4 days, and then you just simply replace the cells. You don't need to change the probe. And the greatest advantage, which when you compare it to chemical analysis, was there was minimal sample pre-treatment. So you could use dirty samples. You didn't need to filter the sample if we put it to a dirty biological sample. However, the issue with this, because you're trying to express as many pathways as possible to consume all your organic acids in sample, there was no specificity. So you couldn't detect a single analyte if there was another analyte present that it could also consume. So building upon that idea, that concept, came up with the idea of the analyte-specific whole cell exclusion biosensor. So what we would do here is we would take a single microbe, single E. coli strain, and entrap it, like it was done with the biological oxygen demand sensors, behind the cellulose acetate membrane onto a dissolved oxygen probe. So this is the cellulose acetate membrane here, and behind that would be the cells. So the difference between the two, this was the old biological oxygen demand process. As part of the PhD in my ongoing work, we genetically engineer a single E. coli strain to exclude all catabolites within solution except for the single catabolite you wish to detect. all pathways within a single E. coli microorganism are being removed that correspond to analytes you would typically find within that biological sample. And at the end of this process, you end up with a single strain that can catabolize a single organic acid. So just as an example, the biosensors that we've developed, we can detect numerous analytes now at present, but to give acetate and propionate as an example, the acetate biosensor, it would have an ability to catabolize acetate, and it would be incapable of catabolizing propionate and a large list of other analytes that are also present in solution. Whereas the propionate exclusion biosensor, it would have an ability to catabolize propionate, but would not be able to catabolize acetate. And again, the long list of other metabolites that are also found in solution. So the genetically engineered cell that will be entrapped on the tip of the probe. It can only catabolize acetate. It has no response from other analytes found. And what you do is you have a concentration-dependent O2 consumption response. So you would use standards to identify, this would be 0.1 millimolar acetate. You would have an O2 consumption response. And then when you put an unknown biological sample in there, you'll be able to map what the actual concentration of acetate is in solution. So the creating of new exclusion biosensors for new bioprocess, new green bioprocess. Initially, they were developed for anaerobic digestion, but upon completion of my biosensor, a lot of the area of focus had moved beyond anaerobic digestion into the green biorefineries. And because I developed these biosensors for grass silage fed AD, the natural progression would be for grass silage fed green biorefineries. So all the steps I've proposed there, this is kind of for every iteration of a biosensor, for every bioprocess we like to apply the biosensors to. We have this phrase, to detect, you need to exclude. We begin with a biological sample, perform extensive chemical analysis in that biological sample. We find all the analytes that are present within that sample. We then look at the strains we have in our catalog, and if there's additional analytes there that we haven't knocked out the corresponding pathway for, we create a biosensor strain, we test it on those analytes, and if there's a response, then we go back and knock out that pathway until we end up with a solution, until we end up with a strain that when we apply it to a biological sample, and we want a single analyte we can detect, all other pathways are removed, what we should see is no response from all the other analytes and just a single response from the analyte we wish to detect. So it's an iterative process. This process is repeated until we end up with this strain here that has a response to a single analyte we wish to detect. So based upon a lot of the work I did in my PhD, I was able to be involved in two projects. One project, the Life Farm for More, which I am the project manager on, the research fellow on this project. I'm developing biosensors for grass silage fed green biorefineries. This is an infographic taken from our project website. You can see the biosensors in terms of green biorefinery issue they have would be the quality of the feedstock and which pathway they want to pursue based on the juice that they produce. And this is why we're developing a biosensor here. The other use of the biosensors, they formed a large part of another project. It's a Marie Curie ITN, project called Agrifine. And in this project as well, a central part of this is green biorefineries, and actually the biosensors we developed here in UCD as providing monitoring control system to that process. So that's everything I'd like to discuss today. Hope you enjoyed the presentation.
Joe, that's been